Okay, so we're going to be starting now. The question I asked everybody, and yes, we are recording, was just to list a few different monomers, as many as you could think of, and see what you all came up with. So, looks like, okay, nucleotides, fatty acids, amino acids, and monosaccharides. Someone must have had the textbook out. Look at that. Perfect. Fatty acids, amino acids, monosaccharides. Oh, nucleotides. Yes, there's 4 different types of nucleotides. That's one. Adenine, guanine, cytosine, and thymine. But yeah, that first one actually hit the nail on the head with what we're going to be talking about here. Is definitely the nucleotides, fatty acids, amino acids, and monosaccharides. So we talked about basic chemistry in Chapter 2. Now it's time to give chemistry meaning, the chemistry that really matters, I always say, the organic stuff. And I'm not talking about the fancy organic stuff from organic chemistry. I'm talking about the first 20 on our periodic table, the ones that are made of those, and the very simple structures, the ones that make up life. Because life truly is not that complicated. If it were, it wouldn't be here. We'll just put it that way. So organic molecules contain carbon and hydrogen atoms. You guys remember your N-CHOPs, right? Okay. And the 4 classes of organic molecules, the biomolecules, if I ask you what they are, I'm of course referring to carbohydrates, lipids, proteins, and nucleic acids. The monomers of carbohydrates are monosaccharides. The monomers of lipids are fatty acids. The monomers of proteins are amino acids, and nucleic acids are nucleotides. Okay? I like this class because you're the first class that has not laughed when fatty acid was said. Usually that induces like vulgarity and laughter. So, yes, we did. That's true. I'm not going to say that on the mic though. Okay. All right. So when we look at inorganic molecules, those usually contain positive and negative ions. We're talking about things being covalently or ionically bonded. Inorganic substances are usually ionic or covalently bonded. Inorganic are going to be usually ionic bonds. Okay? They always contain small numbers of atoms. Usually aren't these big plethora of molecules like C6H12O6. Does that sound organic or inorganic? Organic. As soon as you hear me put numbers after the letters and symbols, you know you're probably talking about an organic molecule. And inorganic are often associated with non-living matter, whereas organic are associated with living matter. I know, I gave you guys a chart and you're filling it out in your notes. I'm aware of that. I'm going to give you a second. You can abbreviate. They don't have to be long sentences. The beautiful thing about organic chemistry is they're going to bond covalently. There's not going to be this oxidation thing that happens. If you remember, ionic bonds are oxidative reactions, redox reactions. What do you guys think? 1 more minute? We good? Roughly? 5, 4. No? Okay. This is an accelerated class. Even time gets accelerated. Yeah. Okay. I can always bring that up at the end if you guys still need to get that down, but I'm going to move on. Okay? So the carbon skeleton and functional groups. We've talked about the various forms before in biology last year or the year before, or if you've had organic, you understand that you can have a straight line of carbon. It can be a tree. It can be even a ring, and we've dealt with some of those in the last lab we did, or the POGO activities. So the carbon chain of an organic molecule is called its skeleton or backbone. So if I refer to the backbone of a molecule, I'm just talking about the line of carbons. It can be a ring of carbons. It can be a path of carbon, so to speak, where it does the little zigzags. But if I say the skeleton, I'm talking about the path of carbons that go down a molecule. And then the functional groups are going to be clusters of specific atoms bonded to the carbon skeleton with characteristics, structures, and function. And it's the functional groups which give that particular molecule its functioning attribute, or what it actually is. There are 20 different amino acids. How many functional groups are there for amino acids? 20, because every time I change a functional group, it becomes a new amino acid. We'll talk more about that too. But it's going to determine the chemical reactivity and polarity of an organic molecule. When I say polarity, do I mean that molecule has a charge? No, it means probably different ends of the molecule are going to be not charged, but contain either more or less electronegativity, right? Just like water. Oh, I'm going to have to zoom in on that. I can tell already. So looking at some functional groups. What? If I look at a hydroxyl group, we've already seen these in the lab we did last chapter. A hydroxyl group, that's going to mean that you're looking at an alcohol. If I see a hydroxyl group hanging off the end of a carbon chain, that indicates to me this is an alcohol. Okay? Alcohol is also polar, just like water. Does that mean alcohol is attracted to water? Yes, it does. You guys remember doing the lab last year, cohesive and adhesion of water and alcohol, and comparing the two? Yep, and alcohol was less cohesive and adhesive. That's why alcohol evaporates, right? Because it doesn't hold on to itself as much. Okay? And then the carboxyl groups we've talked about already. We're going to deal more with these groups when it comes to our amino acids, and we'll talk more about that later. But it also is an indicator of aldehydes, and like formaldehyde in that case. Okay? And then here we have another functional group. This is just an indicator of a ketone. And these R groups. Why does each functional group have an R on it? Can anybody tell me? The remainder of the molecule, but if you asked me, I would say it's because scientists call it an R. Why? Because you look at the periodic table, and is there an R on there? No. Okay? And so I get it's the remainder of the molecule, but I always say, it's my way of remembering, it's not an element on the periodic table, and that's why they did that, so you don't accidentally mistake it, right? And say, oh, it's R. You think that's radon. No, radon is what, RN? Yeah. Okay. So then you have the amino group, NH2. Okay, we understand that's the amino group, and then whatever I attach to that is going to change the amino acid. So in this case, the R could be 20 different things. And we could just go in with examples, but I'm not going to. We're going to move on. So then that leads to something called an isomer. Isomers are basically, if I give you the same chemical formula, or I give you the same amount of elements, and then you all are told to build something, will you all build the same structure? If I all gave you 6 carbons, 12 hydrogens, and 6 oxygens, would you all build the same structure? No. And if they were all different than each other, those would be isomers of the same chemical formula. Does that make sense? Okay. So isomers are organic molecules that have identical molecular formulas but different arrangements of atoms. Often on the AP exam, they will give you, let's say, glyceraldehyde and dihydroxyacetone. Those are just, they could be any 2 random molecules. And they would expect you to be able to go through and go, okay, that has 1, 2, 3 carbons. Okay, 1, 2, 3 oxygens. And that one does 2, 3 carbons, 3 oxygens. And then they have the same number of hydrogens. And you're expected to know that they are isomers. Okay? They aren't going to necessarily give you the formula, but you should be able to look at these 2 molecules and understand that they are the same formula but a different structure. They are isomers of each other. You guys think you could pick that concept out on a test? Okay. So looking at biomolecules, carbohydrates, lipids, proteins, and nucleic acids, those are the biomolecules of life. That's what makes us us, or makes up us. And they usually consist of many repeating units. And each repeating unit we know is a monomer. Now, a molecule is composed of monomers, and that is called a polymer. Can you guys give me an example of a polymer? Why don't you guys type them in? Give me an example of a monomer, or a polymer, polymer. Polymer. Yeah, be specific. Give me an example of a polymer. What is the entry code? Please give me a moment. HUROR. Five of you. Take a guess. It's okay to be wrong. Type it in. Give me an example of a polymer. In 5, 4, I only need one from you. 3, 2, 1. Polysaccharides, yes. Lipids, that's a general category. So not necessarily, we'll say. Fat would have been a good one. Or I guess a triglyceride would be a polymer in that case. Proteins, yes. Proteins contain many monomers called amino acids. Polysaccharides contain many monosaccharides. Phospholipids, yeah, there's more than one. There's 2 in there. I guess that works. See anything different? What am I missing? Did anybody have polysaccharide? Oh, we did that one. Many sugars. Yep. Sorry, there it is. And I'm missing one. What am I missing? Proteins, lipids, nucleic acid, DNA and RNA, right? Yeah, that's the one that everybody always overlooks, I know. So DNA and RNA are also polymers. Don't forget that. So when I look at the monomers of carbohydrates, the monosaccharides make polysaccharides. Lipids, those are going to contain glycerols and fatty acids, and together they make fats, also known as triglycerides, phospholipids. You guys named a couple. I was looking for the specific ones. And then proteins are made of amino acids. They create what's called a polypeptide. If polypeptides fold into a structure, they're called a protein in that case. And then nucleic acids are made of nucleotides, monomers. They make DNA and RNA. Okay? So how do I bring monomers together? Gentlemen, this leads to something called a dehydration reaction. This is something we do not cover in general biology. It's a chemical reaction in which subunits are joined together by the formation of a covalent bond, and water is produced during the reaction. If water is a product, you automatically, know it is a dehydration reaction. If water is in the reactants, you know it is a hydrolysis reaction. Okay? That's kind of how you look to where the arrow is pointing. If the arrow is pointing at water, it is a dehydration reaction. If water is pointing, or if the arrow is pointing away from water, you know it is a hydrolysis reaction. So these are used to connect monomers together to make polymers. So if I look at how you make starch, every time you make starch, which is a polysaccharide, you're bringing a new monomer together and removing water in the process. So hydrolysis is the opposite of that. That's where water is actually going to break down a covalent bond. And this is used to break down polymers into monomers. An example is the digestion of starch into glucose monomers. What happens when you take a piece of Wonder Bread and put it in water? Or crackers in soup. It starts to break apart and dissolve, right? That is essentially a hydrolysis reaction taking place in your soup, or if you're feeding the ducks in the pond bread. Okay? It starts to break that down. So when you see water being removed, in this case a hydroxide and a hydrogen, to make water or H2O, we call it a dehydration reaction. Once that occurs, these two monomers will no longer be separate. They will now be together, and that would no longer be two monosaccharides. It would now be a disaccharide. More than two would be poly. Yeah. Okay. So it can work the other way, where I add water, and that will, again, parts of it will bond with each different monomer, separating it. So if this reaction goes the other way, we say it is a hydrolysis reaction. Let's break the word hydrolysis down. Hydro means what? Water. Lysis means to break apart or burst, right? So this word literally means to break apart using water. So I could give you any type of chemical equation. You must understand which is which. The way I always say to remember, if the arrow is pointing towards water and water is a product, you know it's a dehydration reaction. If water is pointing away, if the arrow is pointing away from water, you know it is a hydrolysis reaction. It's a very simple way to recognize these on an AP exam. You're going to see these, one of these two. I don't know which one, but they're going to give you a reaction. Water is either going to be in the reactants or in the products, and you need to be able to understand and choose the answer that's talking about either hydrolysis or dehydration synthesis. Why do we call it dehydration synthesis? I get dehydration. You're removing what? Water. Why synthesis? You're making something bigger, right? You're synthesizing it. So enzymes are required for cells to carry out dehydration synthesis and hydrolysis reactions. And remember, an enzyme is a molecule that speeds up a chemical reaction. What type of molecule, I'm going to ask you a little question here. What type of molecule is an enzyme? See if you remember. What type of molecule is an enzyme? What type of molecule is an enzyme? All enzymes are this. All enzymes are this, but not all this are necessarily enzymes. We'll say that. Whatever this is the answer I'm looking for. All enzymes are this, but not all this are enzymes. What am I? A little riddle for you. All enzymes are this, but not all this are enzymes. I'm not giving you an example. No, I want the biomolecule I'm talking about. I want the polymer I'm talking about. There's a really big hint. No pun intended, because they're made of many things. 3, 2, 1. Proteins is correct. So all enzymes are indeed proteins. Not all proteins are enzymes, though, right? Okay. So looking at carbohydrates next. Now we're going to be taking a look at the different types of biomolecules. Okay? Did I skip something? I remember it's because I asked a question. So an enzyme, remember, is a molecule that speeds up a chemical reaction, and enzymes are not consumed in the reaction, and they are not changed by the reaction. What does that mean if I'm an enzyme? Can I be reused? Yeah, and that's the cool thing about enzymes. I copy DNA onto RNA to wiggle through a nuclear to go out to make a protein. If this protein facilitates a chemical reaction, it's called an enzyme. And if I had to go through that process every time I needed a chemical reaction to take place, that means every time I wanted to do a chemical reaction, I'd have to make a new enzyme. There's too much energy. Okay? So we're able to reuse enzymes over and over and over again. There's one enzyme in your liver. It helps break down hydrogen peroxide. It's called peroxidase. If you leave hydrogen peroxide in your bloodstream, it's toxic. Yes? An enzyme is a catalyst by its definition. But a catalyst doesn't necessarily have to be an enzyme. A catalyst is just anything that speeds up a chemical reaction. A catalyst could be friction striking a match, the heat caused to make that chemical reaction occur. But an enzyme, remember, an enzyme lowers the amount of activation energy for a reaction to occur. Remember that definition from biology? Okay, so a catalyst can do that too, but if I am a protein and I do that, I am now an enzyme. Do you follow that train of thought? Okay. Now I've lost my train of thought. So no, but you guys, enzymes in the body, like the catalase that I just talked about, if you had to literally make that protein / enzyme every time you had to break down hydrogen peroxide, you'd die of hydrogen peroxide being in your bloodstream. You couldn't do it fast enough. So that enzyme facilitates the reaction and then can do the same reaction again. Do enzymes get wore out? Yeah, and then you make new ones. Can your environment change enzyme shape? Yeah, and then you can destroy them, right? And you can wreck their shape. And if you wreck an enzyme shape, it can't function anymore. Structure equals function. So looking at carbohydrates, and we're going to talk more about those. There's a pogo we're doing in this chapter over enzymes. So looking at carbohydrates, we understand that they're a source of our energy. We get that. They provide building materials. It's our structural role for some things, like plants especially, right? Cellulose. Okay. They contain carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. What is in a 2 to 1 ratio that makes up 70% of us? Water. Let's look at that word. Carbo means what? Carbon. Hydrate means water. So this is 1 carbon with a 2 to 1 ratio of hydrogen to oxygen. Did you guys get that? Think about it. C6H12O6. Carbon in a 2 to 1 ratio of hydrogen to oxygen. It's right in the word. Carbohydrate. So it contains carbon, hydrogen, and oxygen to that 1 to 2 to 1 ratio. If I gave you a molecule on a test and you counted carbon to hydrogen to oxygen, even if I gave you C12H24O12, you would know that's a carbohydrate. More specifically, it's probably a disaccharide, right? Okay. So the varieties are monosaccharides, disaccharides, and then, of course, our polysaccharides. We talk about disaccharides because glucose can form several different disaccharides based on the different isomers of glucose, and we're going to get into that a little bit later. What is a disaccharide that we see all the time? I heard of sucrose, table sugar, right? Okay. What is table sugar? It's glucose and another one called, it's a little fruity, fructose. So looking at our monosaccharides, it's a single sugar molecule and also has simple sugars. If I ask you what a simple sugar is, you would say that's a mono or disaccharide. It has a backbone of 3 to 7 carbon atoms. If I'm glucose, I have 6. Some examples of those are glucose, that's in our blood, fructose, that's a fruit sugar, and galactose, which is known as grape sugar. That's what a lot of scientists label that one as. But these 3 monomers are basically what are going to make up all of your other polysaccharides or disaccharides. So if I say it's a hexose, it's a 6-carbon sugar. Go figure, right? Makes sense. Hexagon, we get that. And if I'm a 5-carbon sugar, like in my nucleic acids, that's a pentose sugar. Might they use that language on a test? Yeah. What types of monomers have pentose sugars? You would tell me nucleotides. Do you see how you're expected to think about this now? If I gave you a question like what monomers contain pentose sugars, you'd automatically go to, 5-carbon sugar in my nitrogen bases, right? And I know that there's either ribose or deoxyribose in nucleotides. And so that's where my brain automatically goes. That's pentose sugars. If you see hexose, you're probably talking something like a sugar or a carbohydrate in that case. Okay? Don't worry if your brain didn't automatically go there. Okay? But that's what I'm trying to get you to think like in this chapter. Where if you see a pentose, you know that's a 5-carbon sugar, and then you remember from DNA that it's made of 5-carbon sugars and its nucleotides, right? Okay. So glucose, again, we see our hexose sugars, C6H12O6. These are 2 different types of this is glucose drawn, I should say, in 4 different models. Do you see how each different model gives you more or less detail? Remember the last lab we did? Yeah. These are all exactly the same thing. These are all a glucose molecule drawn different ways, each giving you more or less information. This one I have to automatically infer that my carbons and hydrogens are hanging off the ends of these particular molecules, right? We all good there? These guys right here? I'm sorry, what? Well, oxygen, this oxygen's happy here. We know that actually has 2 bonds. And in order to fulfill, which ones you talking about, the OH groups hanging off? You would have to have those, otherwise you wouldn't have all of the oxygens in the actual molecule. So if you can't automatically get to here in your head, go back to here, okay? Because you would need to fill in all of your oxygens and your hydrogens to fulfill C6H12O6. Does that make sense? Okay. I know. My brain doesn't go to glucose. Even me, I don't go to glucose right from here. I go to glucose from here. Okay, this model B would be enough for me to say, oh, that's glucose. I would have to work back and draw even C out to understand that was glucose. But based on what the molecular models we've looked at so far, we need to understand that you can get to here with just this equation. That's what that lab was trying to instill with you. Not can you do it, but can you visually understand how it's being represented by this carbon ring. Oh my goodness. So that leads to disaccharides. Hello. I do. Okay. Bryant, somebody's here to pick you up. So looking at a disaccharide, di we know means two, saccharide referring to sugars, in this case 2 monosaccharides, Duffy. So this contains 2 monosaccharides joined together by dehydration synthesis. What is dehydration synthesis? Removing water molecule and bringing 2 things together. So in this case, this is the 3 times with different monomers of sugar. You can either have lactose, sucrose, and maltose. We talked about sucrose already. These are the 3 disaccharides you're expected to know. Lactose, this is known as milk sugar. This is what you find in your milk that you drink every day. Okay? It's composed of galactose and glucose, basically grape sugar and glucose sugar. I will tell you that all 3 of the disaccharides are going to contain glucose. Then there is sucrose. That's the one that we are most common with. That's table sugar. That's composed again of glucose and fructose. And then there's maltose, which is like super glucose, right? It's glucose and glucose. What's that thing that people drink? It's called a what? Malt, right? A chocolate malt. And it's super what? Sweet. And that's why. It's like double glucose. It's what your body wants times 2. Okay? Because that's why it tastes so good to our taste buds, really, if you think about it. So then we have synthesis and degradation of maltose. So how are we going to break it down? Well, we know we break this down by hydrolysis, right? Okay, so we're going to break it down using water. And if you put sugar in water, we know what happens. So in this case, when I break down maltose, it will break down into glucose and glucose, 2 glucose molecules. These 2 will be identical to each other. You say, wait, they don't look the same. Yeah, but we know over here there's an OH group hanging off the side. How do we know that? It's the only way these bonds would be satisfied. And if we jump back, just to remind you, there it is. Okay? Remember, glucose can look like this, it can look like this, it can look like this, and it can look like this. I really like this model best. But you don't always get that model. So if I'm going to break this down, or bring these together, to make a maltose, in this case I know I'm going to have to take the hydroxide group and the hydrogen and remove them to make water. In doing so, I will go through a dehydration synthesis reaction. And this oxygen that's left over will have no choice except to bond where the hydrogen was removed, because it wants to bond how many times? 2 times. Remember, this is all about just fulfilling that outermost valence shell or octet, right? So we know oxygen wants to bond twice. It's going to do so because water was removed and that hydrogen was left a void, and the oxygen and hydrogen that were removed from the other molecule left a void. That's going to be fulfilled by the leftover oxygen. And water will be a product. If the arrow is pointing at water, it is a dehydration synthesis reaction. So if this is, oops, sorry, I just talked about all that. I apologize. So if I go the other way, now I have maltose. Now I have a disaccharide called maltose. I'm going to break it down. If I add water to the situation, am I looking at reactants or products at this point? Reactants, right? Okay. So if my arrow is pointing away from water, what type of reaction is this? Hydrolysis, right? Okay. So now I'm going to add water to break this molecule down. It's called a hydrolysis reaction because water is being added to a disaccharide to separate it into 2 monosaccharides. In this case, it's maltose is the disaccharide, and the 2 glucose is my monosaccharides. Okay? There's no water in the product, so I know it's a hydrolysis reaction because the arrow is pointing away from my water molecule. Okay? Are there any questions about dehydration synthesis or hydrolysis? If you have not had chemistry or organic chemistry, this is a new concept. Okay, I know that. So some of you who are not there yet, as juniors, I know are taking chemistry, this is like, whoa, Mr. Mason, that's a lot all at once. Don't worry about it. This is part of the class, should be just fine. Plus the chemistry you're about to have is going to reinforce what we just talked about. So these reactions can go either way, which gets kind of confusing from time to time. What's the rule I tell you? If the arrow points away, away from water, it is hydrolysis. If the arrow points towards water, it is dehydration synthesis. So that leads to polysaccharides. What we just looked at is making one disaccharide. Now, how do I get many together? Well, a polysaccharide is a polymer of monosaccharides. And the examples I give you are starch, which provides energy in plants. We understand that. Your potato has a lot of what in it? Pasta has a lot of starch in it, so does potatoes, okay? And then there's glycogen. And this is like an animal starch. Do we have glycogen? Yeah. Now I'm going to ask you a question. What types of tissues in your body would you say have a lot of glycogen in them? What types of tissues in your body would have a lot of glycogen in them? Yeah. What types of tissues? Just take a guess. I don't want to hear you talk. I want you to type it in. It's okay to be wrong. Give me what you got. Put it in the machine. What? I'm sorry, what? I know. In just a little bit. So let's see. We'll pick up from here tomorrow, but I want to see how close you are. Yay, muscles, perfect. Glycogen, adipose. Adipose is not a bad guess, but we just don't do it there. We store it differently in the adipose tissue. We'll talk about that later. But muscles is really what I was looking for. Your ear. We'll put ear on there. No? No one's going to fess up to that one. Okay, so muscles. So if I was a turkey, what type of meat on a turkey do you think has more glycogen in it? The dark meat, right? Because it's flying all the time. They have to store lots of energy there. So I'm going to stop right there. It's kind of a fun question for the end. Yeah, I can go back. I'm not done yet anyway. So tomorrow we'll pick up where we left off.